United States Department of Agriculture.
Good morning. I guess in Philadelphia, I don't know where everybody is. My name is Mohammad Kiani. I'm at Temple University in Philadelphia, United States. And what I want to talk to you about today is some of our effort that's been going on now for close to 20 years in trying to kind of come up with a comprehensive approach to identify therapeutics for treating sepsis, which, I think the previous speakers have talked about the importance of this. My main collaborator in this is Dr. Laurie Kilpatrick at the medical school here at Temple University. So, as I said, our objective has been to develop a comprehensive framework to understand the inflammatory response and screen therapeutics for treating inflammation, specifically when it comes to sepsis, has been our disease model. So we have been using a synergistic combination of organ-on-chip, omics, clinical data, and in silico models to understand sepsis. And the idea here is that we go from animal models into organ-on-a-chip, into an in silico model, essentially a mathematical model, and then come back to human to see, how this whole thing works. And so I'll talk a little bit about each of these components and see at the end, hopefully you'll see how these things come together. In terms of the general idea of inflammation, I think I probably don't need to do this for this audience, but in general, what happens in a healthy endothelium, you don't have neutrophils do not interact very much with endothelial cells. So we don't have an inflammatory response. However, when endothelium is activated during diseases such as sepsis, two things happen, two distinctive components happen. One is there's a molecular change, upregulation of various molecules, cytokines and chemokines and things like that in the tissue. But there's also a change in the way blood flows and the fluid dynamics of the system. Neutrophils become more marginalized into the tissue. And so there's a microfluidic component to this as well. And of course, this has been used, animal models have been used to study these things for a long time. And as you will see, this has been our starting point as well. And so what we have done is, you'll see, I'll talk about this in a second for some of the idea, but we have developed an organ-on-a-chip microfluidic system that mimics the microphysiological conditions, allowing us to observe real-time imaging of leukocyte rolling, adhesion, and migration, all on the same system, discover role of shear forces, mechanistic understanding of leukocyte and endothelial interaction. And then this has allowed us to predict in vivo responses of various therapeutics. So how did we do this? We started with an animal model. You see this in this one in the left upper corner. This is from actually a microvascular network in an animal. And we developed a long time ago, this is more than 20 years ago, we developed a methodology for mapping the complete mapping of these microvascular networks, including all the fluidic components and all the various measurements and things like that. Then this was put into a GIS system. This is the similar type of system that you use for GPS systems on your phones, probably. And then this graphic here shows how we map this on a chip, essentially reproduce this complete microvascular network on a chip, including various tissue sites. And so we could get a situation where the tissue component is connected to the vascular component through 3 micrometer gaps, which is the standard of what's been seen in vivo. And this allows us to look at rolling, adhesion, and migration of neutrophils in the same chip. And so this, again, kind of shows you that the overall design. This is the complete microvascular network reproduced on a chip. Then you see the neutrophils here flowing. And then this is an image of how the system looks like. And you see that the tissue compartment, you see the vascular compartment and the gaps, how they're connected together. The advantage of this system, or one of the advantages of the system, is that it gives us a complete view of the system. You are not looking at an individual vessel. You're able to reproduce the complexity, for example, of the shear rate values in the system. You could look at bifurcations. You could look at complete networks. And again, it's very close in terms of its geometry and structure to the in vivo system that we started from. And so this model has been validated and has been published for a long time. If you're interested, contact me. I can send you some information. And we have compared this. We have shown that it compares well with animal models and also other existing models. And so I'm not going to talk too much about the validation, but just to give you a sense, this is when we cellularize this, so we can put endothelial cells in the vascular compartment. And when we cellularize it, completely covers the whole system. And here is a video showing how this system functions. So you can see the vascular channels. And now you see fluorescent neutrophils coming into the system. And you can see them coming in, going to different vessels of the system. This kind of looks like traffic in Philadelphia once in a while, but you can see how this goes, and you can see some adhering neutrophils now. And then, again, you can see different shear rates, different flow rates in different vessels as the system functions. And then, some of the neutrophils are already adhering. Some of them are starting to migrate. Of course, the migration takes much longer, but you can see that they are starting to get into the tissue compartment. And so this kind of gives us a very realistic view of what happens in vivo. We have compared this to the in vivo system, and comparison is very good. The correlation is very good between the two systems. So we see the same kind of flow parameters, adhesion, migration patterns that we see in the matching system in vivo. And even if you look at it at high magnification, I don't know how well you can see this image, but you can see a neutrophil migrating between the poles into the tissue compartment here. And it's kind of creepy, but you can see it moving, and then you see that in a second it's going to change its mind and it's going to come back a little bit. And this is very much consistent with what we see in vivo. So again, just to emphasize, that's not the point of this presentation, but to emphasize that what we are seeing is very realistic and very reproducible. So what can this be used for? We have used this for a number of different applications, but just a quick example that might be of interest to this audience is this idea of whether what you see in one species is reproducible in another species. And so, for example, we wanted to see whether neutrophil endothelial crosstalk is comparable between mouse and human cells. So in this case, what we did, we tested the response of a compound called PKC delta inhibitor. It's a promising compound for treating sepsis. We wanted to see how this compound responds if we put human cells versus mouse cells in our system. And here is an example of this. You can see, first of all, on the x-axis, you can see that these are different shear rates representing different vessels, essentially. So we have vessels with low flow, we have vessels with medium flow, we have vessels with higher flow. And then we can also look at bifurcations. And this is in the case of human, you can see that with no treatment, there is low number of adhered neutrophils. When you upregulate this with TNF alpha and FMLP, you see that, as expected, the number of adhered neutrophils goes up. And then when you treat it with this PKC delta inhibitor, it goes back down. And this is true of low flow vessels, but not so much at bifurcations or that the total is responsive. If you look at the case of the mouse, you kind of see a similar response when it comes to adhered neutrophils. So this is a summary graph at the bottom. You see that they are relatively comparable. The response of mouse cells and human cells for adhesion is comparable. However, if you look at migration, we see a different picture. You see that in terms of the migration over time into the tissue compartment, again, for no treatment, it's slow. When you activate the tissue, the chip, the number of migrated neutrophils goes up. But then when we treat it with PKC delta, it goes back down to the control level. And you see that this is very dramatic for human cells. However, if you look at the case of the mouse on the right-hand side, mouse cells, you see the same thing for the no treatment and TNF treated. But you see that even though PKC delta is effective in reducing migration, it's not as effective as it is in human cells. And so this is a very important thing. Imagine all the cases where a drug like this would be, let's say, tested in mice and would be probably maybe abandoned because the response is not good in a mouse model. But we wouldn't know that this might actually work very well in a human model. And so this is very important to show that, just because something works or doesn't work in mice doesn't mean it necessarily corresponds to what you see with humans. And so we've gone through this. Work is published. If you're interested, let me know. I can say that. But this is an important distinction that you can imagine how many drugs were abandoned because they didn't work in mice and they might have worked in humans. And then this is, for example, the number of adhered microparticles. This is for, if you look at the adhesion molecule upregulation in the system, and you can see that the E-selectin is similar between mouse and human, but ICAM is not. ICAM is upregulated in human cells, but not in mouse cells. And so this is another important distinction that this kind of system can provide for us. We can also look at another version of this. We can look at brain during inflammation. This is a brain model of our system where we can grow brain primary microvascular human brain endothelial cells in this model. And then we can, in the tissue compartment, we can put astrocytes and pericytes. So this is a multi-culture system. And you can see actually the astrocytes are forming end foot here with endothelial cells, which is what you would see in an animal model. So we can reproduce that phenomenon here. And just a quick, again, this work is published, but just a quick summary of what we see in here. So for example, if you look at permeability in this system, the permeability is low under control condition. It increases when you treat it with TNF. And when we treat it with PKC delta inhibitor, the inhibitor I was talking about, the drug I was talking about, it goes back down. We can also measure TEER, electrical resistance, essentially, transendothelial electrical resistance in this system, and which of course is reversed. The resistance is high in the control case. It goes down with TNF, and then it comes back up when we treat it with our therapeutic. So again, you can use this model to look at what's happening in the brain and so on. We have a tumor model of this and so on. So, this has been well developed. In the brain model, we can, for example, look at adhesion. And again, you see the same phenomenon. Adhesion is low under control condition. Adhesion is higher in the TNF alpha treated, but then it goes down. Same thing with migration. And there are various conditions. You can see that we can show that this therapeutic, this inhibitor, works well in reducing migration into the brain parenchyma. So these were just some quick examples, but what we want to do is whether we can use this to screen various therapeutics. So this data is unpublished. It's not published yet, but you can see in here, we wanted to compare two different drugs for treating sepsis. One is this BN52021, which has been used in clinical trials that failed. It failed in clinical trials. The other one is this therapeutic that we've been working with, this PKC delta inhibitor. And you can see some interesting results here. If you look at FMLP as the chemoattractant that you use in your system, you can see that the response of the BN and the PKC delta inhibitor are different, right? You can see that the PKC delta inhibitor is a much more effective in treating response to FMLP, both for adhesion and migration. However, if you use IL-8 as your chemoattractant, then you see that the two BN52021 and PKC delta inhibitor work the same. The response is the same for both of them. It's similar, I shouldn't say the same. The response is similar for both of them. And what this tells you is that our system probably could have predicted that BN52021 is not as good of a treatment for sepsis as something like the PKC delta inhibitor. So this system would be useful for determining the degree of effectiveness of a therapeutic before you get into expensive animal and even human trials using these kinds of drugs. And by the way, the BN521 worked well in animals. It just didn't work well in humans. So having said all of these examples, now I want to get into the main part of this, which is how do we translate this into clinical applications? So we know that new approaches are needed to better translate findings from various models to human disease. And so we have used a synergistic combination of approaches, both in vivo, in vitro, omic, and in silico modeling to see how the system works. We already talked about how important sepsis is. There is, 47 to 50 million people every year treated around the world, at least 11 million deaths per year, 1 in 5 deaths worldwide associated with sepsis. But the sepsis 3 definition, which is the accepted definition now, consensus definition, is that sepsis is a clinical syndrome that's defined as a life-threatening organ dysfunction, and it's caused by a dysregulated host response to infection, essentially an overreaction to the infection. So what is the current state of treatment for sepsis? Mostly antibiotics and supportive treatment. There is no specific pharmacological treatment for sepsis. There has been something like 150 drugs that have been developed in the past 15, 20 years in animal models, and all of them have failed in clinical trial. So none of the drugs that have been worked in animal models have succeeded in clinical trials. Patients with sepsis often die of organ failure. Again, neutrophil dysregulation and neutrophil endothelial cell interactions have a critical role in organ damage. So we need new approaches. And this failure of animal models got so bad that New York Times even at one point got into this game and said much time and money has been wasted studying mouse models of inflammation. And so this is a big deal now, at least in the United States. Some of the National Institutes of Health, for example, the NIGMS, do not support animal model studies of sepsis anymore. And so this has become a big issue in the US. So what's going on with sepsis? So recent studies have shown that sepsis is a heterogeneous syndrome. Different people respond differently to sepsis. And so you need to look at the different phenotypes. However, people so far have only looked at transcriptomic biomarkers of immune cells to phenotype patients. No studies have examined neutrophil functional phenotypes in sepsis, how neutrophils react in terms of function. And so this is what we have done. So we used our organ on a chip to classify distinct sepsis neutrophil phenotypes using patient data and microfluidics to see what happens. So our study population was a group of sepsis patients admitted to Temple University Hospital. We had 45 sepsis patients that we looked at in this study. On the organ on a chip, we used human lung microvascular endothelial cells cultured in our system, and then cells were treated with buffer or cytomix. And cytomix is a combination of cytokines that has been shown to well reproduce what happens in patients. Then neutrophils were isolated from blood samples, and then we looked at their adhesion and migration in our system. Then we did an unbiased label-free global proteomic analysis of these neutrophils as well, so that we could match the two groups, the two measures. One is the functional measures from our microfluidic system, one is the proteomic results. These were some of the early patients that we looked at and kind of actually were the ones that piqued our interest. So if you look at controls, of course, you see that when there is no treatment, there is lower level of adhesion. When you activate it with cytomix, there is more adhesion. We already know this. This is nothing surprising here. If you look at patient 4, you see that under neutrophils from patient number 4, when there is no treatment, they are at a low level of adhesion, as you would expect. However, if you treat it with cytomix, patient 4 suddenly shows a very rapid and high response to adhesion. So meaning that a lot of neutrophils start attaching. However, if you look at patient 6 doesn't care. Under control conditions, it has low adhesion. Even under cytomix conditions, activated conditions, it's still low. And the same thing, even more dramatic, is when you look at migration. Patient 4 has a very rapid response and very high response to cytomix. It shows a lot of migration. Neutrophils from patient 4 show a lot of migration, while neutrophils from patient 6 don't care. They don't migrate. And so these two patients, as far as the clinicians are concerned, both of them have sepsis. There's no distinction between them. However, if you treat both of these patients the same, which is what they do right now, you probably don't want to give patient number 6 an immune suppressant because their immune system is already at a low level. You don't want to suppress patient 6. Patient 4 probably could benefit from that. And so we started looking at this. And by the way, both of these had acute hypoxic respiratory failure, and they were both on mechanical ventilation. So as far as the clinicians were concerned, these patients both had the same symptoms. So when we looked at all these patients and we did a discriminant analysis on the results of all the results, adhesion, migration, all the results we could get, the patients naturally siloed in 3 groups, which we called hypoimmune, hybrid, and hyperimmune. And so if you look at the data, how these patients segregate, if you look at the number of adhered neutrophils, you see that when we treat the neutrophils from these patients with buffer, there is no difference. They're all the same. However, when we treat them, when we activate them with cytomix, you see that the hypoimmune group, the group that we call hypoimmune, has very low levels of adhesion, while the other groups, including the control, have relatively higher levels of adhesion when they are activated. So this hypoimmune group has some kind of a low response to activation. If you look at the migration of neutrophils, you see the same thing. Again, with the buffer, we don't see much of a difference between the groups, right? With the buffer, they're all the same. They're all at a very low level of migration. However, when you activate these neutrophils, now the picture is a little bit different. The hypoimmune is low again, but so is this hybrid group, while the control and the hyperimmune group show significantly higher levels of migration into the tissue compartment. And so this type of analysis allows us to segregate the group. So I'm just going to go back one slide so you can see what the difference was between adhesion and migration. And so this kind of gives us a nice way to segregate these patients into using functional measures. But okay, so that's the functional measures. Is there a correspondence to proteomics? And there is. I'm not going to show all the data. Again, this data is published, but I'll just show a quick summary. If you look at the overall proteomic analysis, you see that the sepsis patient phenotypes, the functional phenotypes, have distinct and unique proteomic signatures. So you can see that the hyper and the hybrid group have much more similar. This is kind of a global picture. If you look at our publication, you see more details about this. Have more similar proteomic responses, while the control and the hypo group, remember, hypo group is the one that's not non-responsive to adhesion and migration, have similar proteomic patterns. And so, again, I want to emphasize this point again. I already said this. Clinical parameters alone cannot differentiate between these distinct neutrophil functional phenotypes in ICU patients. They all pretty much have the same parameters that clinicians use to group them. There is no way to separate these three groups of patients from each other based on the currently existing clinical parameters. However, if you look at these three distinct functional phenotypes in ICU patient sepsis, they are correlated with disease severity. So you can see, for example, the number of neutrophils in the hyperimmune group is much higher than the other groups. The oxygenation level is, again, different between the three groups. This time it's higher in the hybrid groups. The mechanical ventilation, the patients who tend to be on mechanical ventilation are much higher in the hyperimmune and hybrid group compared to the hypoimmune group. And if you look at the length of stay, you see that this hyperimmune, again, this hyperimmune group has much higher length of stay in the ICU. They don't do as well. Now, in terms of the ICU mortality, we could not see a statistical difference, but you can see that the number of mortality is a little bit higher in the hyperimmune. We think this is because of our small sample size. We think if we collect additional samples and work on additional samples, we should be able to, this will resolve, and we think we are going to see higher ICU mortality in the hyperimmune group. So, in summary, if you want to look at how this platform identified these patients, you can see that in the hyperimmune phenotype, we have higher neutrophil adhesion, higher neutrophil trafficking into the tissue compartment, and we have higher nets formation. I didn't talk about nets, but again, this is published, so I'm not going to go into it. These patients, these neutrophils from this group of patients tend to produce more nets. These patients require ventilation. They have a longer stay in the ICU. Their plasma nets are higher, and they have larger number of systemic neutrophils. In the hypoimmune group, in contrast, the neutrophils are happier. They don't care. They don't adhere and migrate as much. These patients tend to require less mechanical ventilation, and they are in ICU for a shorter period of time. This hybrid group, which is kind of a mix of the two, they tend to have higher adhesion, but lower migration. So it's kind of a combination of the two. That's why we call it hybrid. And they require more mechanical ventilation, but their length of stay in the ICU is lower. So this is an interesting group. that's kind of in between. You're still trying to figure out what's happening with this third group. But this distinction, as I said, has worked well. And remember, there's clear proteomic signatures for each of these groups, or each of these phenotypes, I should say. Okay, so having done this, both in terms of identifying these functional phenotypes and also the proteomic signatures, we are now going to use this to see if we can use mathematical modeling to identify druggable targets and repurpose FDA-approved, Food and Drug Administration-approved drugs for treating inflammatory diseases. Of course, if you can repurpose a drug, it's much easier. We don't require a whole new set of extensive clinical trials for this. It's much cheaper and faster to repurpose a drug than to develop a whole new drug. So how do we do this? What we do is we look at our omics data, we look at the differentially expressed proteins, we look at gene ontology, and then we look at the PPIs and clusters and use standard techniques to develop pathway analysis for various systems. And then we look at these nodes of interest, then we combine this with our machine learning based optimization. And then we use this to look at drug banks to see which of these drugs can impact these gene or protein signatures that we see with our various phenotypes, for example. And then we use this to develop disease and drug pathways. And then we ask this question, are these proteins druggable? Are these pathways druggable? And if they are there any existing drugs that can impact these pathways? So here is an example that we are working on. This is pathways and processes that are with differentially expressed proteins in our system, as I showed the data for, that are targeted by various FDA-approved drugs. And of course, this is, a detailed and complicated kind of a pathway analysis picture. But what I want you to focus on is the fact that if you look at the different pathways, you see that some pathways are only impacted by one type of, or I'm sorry, I should say the other way. Some pathways exist only in one type of phenotype, some exist in two types of phenotypes, and some exist in all three phenotypes. And what this does for us, it gives us an opportunity to come up with drugs that are specifically targeting either one phenotype, two phenotypes, or all phenotypes. So it kind of gives us a situation where we can kind of essentially tailor our approach. And you can see that some of these, for example, neutrophil adhesion migration, cytoskeleton, and some of these ones that are important, netosis and things like that, are the ones that we are looking at in terms of these pathways. So using this type of pathway analysis, we have come up with some examples of FDA-approved drugs and small molecules that can target this neutrophil adhesion migration using our pathway analysis. And you can see, depending on which functional phenotype you're interested in, you can choose your drug or compound and decide, which upregulated or downregulated proteins you want to look at. And then you can look at neutrophil pathways of interest, right? What we know about these things, you can go through and systematically do that. And then you can also know something about the mechanism of action, at least what's known about the mechanism of action of these things. So you're already starting with a system where if you decide to try a drug, you can then, you already know something about the mechanism of action for this drug. So now you have a list of drugs. What do you do with these drugs, right? That's an important issue and next step. So what you could do then, you could get these drugs, you could go back to our microfluidic system, test these drugs on neutrophils from these patients, and at least get a sense of whether this drug that you're interested in or this compound that you're interested in has any chance of addressing the problem of adhesion and migration and neutrophil dysfunction in this kind of system. So you can complete the circle, and this will give you more confidence before you go into animal or even directly into some clinical trials. And by the way, FDA now in the US at least allows the use of this type of data from microfluidic system instead of animal models. So you don't have to have all animal model data. You could use this type of data to go and, as justification for clinical trials. So in summary, this is what we are trying to do. We want to get data from various omics, genomics, transcriptomics, proteomics, metabolomics, Then we get data from physiological changes using our microfluidic system, systemic inflammation, activation of endothelial cells, leukocyte trafficking, vascular barrier dysfunction, permeability, and things like that, which I briefly mentioned in the example from the blood-brain barrier model. We also get clinical parameters and functional measures, such as respiratory rate and PO2. We put all of this into an in silico model, essentially a mathematical model. We start with the gene and protein list. We look at the strings and KEGG and things like that. We look at protein-protein interaction networks. We identify biological processes. Then we use machine learning to identify disease-causing genes and proteins. And then, as I said, we can validate these in our microfluidic system to make sure that these pathways that we discovered are reasonable and work with our disease of interest, in this case, sepsis. We can then put all of this into a machine learning system, combine it with a list of FDA-approved drug candidates, and come up with a patient immune phenotyping system where we can phenotype patients on the go, hopefully, and then use this as a method for precision medicine, essentially coming up with a way where we can treat this. And from the literature, we know that it is not likely we are going to find a single agent that can treat all cases of sepsis, for example. We are going to have to do what's called precision medicine, essentially figuring out what works on which patient. And we think this comprehensive approach will give us a sense of how to do this, or a platform for doing this. So having said this, I want to acknowledge the work of a number of our colleagues in various places. Our clinical collaborators at the School of Medicine, Dr. Marchetti and Dr. Merali. And then we also have collaborations with CFD Research Corporation and SynVivo for our microfluidic work and for our modeling work. And of course, all of this work is done by students and fellows. You know, professors like me, we are just sitting there looking at the results. But the hard work and the real work is done by students such as Dan and Roman and people like that who are doing the real hard work. I want to acknowledge their work. Our work was supported by the Defense Threat Reduction Agency and the National Institutes of Health. And I'd be happy to answer any questions you may have.
USDA is an equal opportunity provider, employer, and lender.